in a certain way, I'm actually very surprised to be here, giving one of a series of public talks that I've given on synthetic biology over the last year or so. When we launched this project, we envisioned it, frankly, as a kind of old-fashioned project for us, a purely scholarly project. Our charge from our funding organization, the Sloan Foundation, was to contribute to the scholarly literature on the ethical issues associated with this field known as synthetic biology. And so I was envisioning that this was going to be a very nice little project, and I'd get to do some interesting work, but it would sort of be under the radar in terms of sort of public impact. It would have a scholarly impact, but it would possibly come and go without people knowing very much about it. But that all kind of, everything kind of went crazy in May of this year when we were having the third of our 3 research project meetings in this project. During the meeting, I got a call from Joe Palca of NPR saying that he wanted to talk about this major new announcement that he'd heard about. And I didn't know what he had in mind, but it quickly turned out that it was this announcement from the J. Craig Venter Institute, a privately funded institute with offices in Maryland and California, that they had succeeded in creating what they claimed was a synthetic cell. They had synthesized a bacteria known as Mycoplasma mycoides, which has a very small genome, about 900 genes, and they had inserted that synthetic genome into a cell from another species, a closely related species, Mycoplasma capricolum, which had its genome removed. And then they'd ended up with what cells that were alive and appeared to function like Mycoplasma mycoides, and then went on and produced generations and generations, millions and billions of cells. So they claimed in their major paper on this that they'd created a synthetic cell, that they'd created synthetic life. They'd claimed that they'd created a synthetic species because I think the theory was that they had modified the genome a little bit. They hadn't just synthesized this genome, but they had inserted some code that stood for J. Craig Venter and I think an email address. And so it was a little bit different from the naturally occurring Mycoplasma mycoides, and so they called it a new species. And this was billed as both an enormous technical leap forward and as a major philosophical advance. And I'm going to comment as we go along on both of those claims. So it's the new IT technology. And I should say, actually, I realized after I'd put together this slide that this comment from the science writer Michael Spector, an article in the New Yorker, looks like it's about the JCVI announcement. It isn't, actually. This was an article that appeared in 2009, some months before the JCVI announcement. So the JCVI announcement really brought this field to public attention, but there's been a tremendous amount of excitement all the way along about it, which both of these quotes capture a bit of. It sometimes seems to us here at the Hastings Center that major new revolutionary technologies or technological advances come around every couple of years or so. There's the Human Genome Project in personalized medicine. There's genetic engineering, gene therapy, stem cells, neuroimaging, nanotechnology. Each one of these is attended by tremendous hype, and then the time goes by, and people realize, yeah, we're actually still fighting Richard Nixon's war on cancer, from the 1970s. But maybe I'm paying just a bit more attention to this one because I'm right in the midst of it and really reading all the literature. But there seems to me to be a tremendous amount of hope or hype surrounding this one. The folks working on this really feel that they are bringing about what is going to be and effectively the second industrial revolution. The first one was sort of petrochemical based, and now they're coming up with entirely new ways of producing all these things that we've been wanting all along, and coming up with new products to produce as well. There'll be huge macroeconomic consequences to it, as this quote from Craig Venter illustrates. And it's going to change our lives in a deep and fundamental way. One of the extraordinary claims that's made about synthetic biology is it's going to bring the capability to engineer life and create new forms right down to the kitchen counter. This is sometimes referred to as DIY bio, do-it-yourself bio. And people will have DNA synthesizers and centrifuges and bioreactors right on the counter. But that's not what this is. This is a picture of my own kitchen, and I offer this slide sort of by way of disclosure. We're often struck here at the Hastings Center that debates about where new technologies, where they're going to go, whether they're good things or not to be doing, often devolve into the two sort of broad camps between sort of the enthusiasts, the ones who get really, really excited about it and kind of go crazy, and the critics or the hand-wringers, as we sometimes call them. And it often appears that this difference traces back to underlying differences in our views about what the human relationship to nature really should be. Some people are drawn toward engineering, toward building, toward creating, toward using resources and making the most of ourselves and the world around us. And some people are drawn more toward accepting human bodies as they are, cherishing and preserving the natural world as we find it. They're drawn toward a stance of gratitude, as my colleague Eric Perrins puts it, toward nature, or toward human nature. I'm not immune to that sort of engineering mindset, but I'm a little bit more in the second camp. What really counts as cool biotechnology for me is a kitchen compost bucket. And that's what this is. So by way of disclosure, I don't find this all that cool, but I'm going to try and do the kind of thing that we do at the Hastings Center. I'm going to try and give you an honest appraisal of the technology, and I'm going to pay attention to the deeper, more problematic moral concerns that have been raised about it. Now, oddly enough, one of the things that's contested within synthetic biology is just what the heck the field actually is, what synthetic biology really means. Some think that it's, some in the field think that it's really just an advanced form of recombinant DNA work, of the kind of genetic engineering or gene therapy that's been around for some decades now. It's quantitatively different. We've got better tools, we've got much more information, we've got more automation. In particular, we've got machines known as DNA synthesizers, which you can think of as effectively a kind of printer. You key in the genetic sequence that you want, and what it prints out is that genetic sequence. And this has spawned a whole industry around these things. So quantitatively different, but not qualitatively different. As the science advisor to our project has put it, he's of this mind. He thinks it's basically the same thing we've been doing all along. He just calls it genetic engineering on steroids, or jet-powered genetic engineering. But others are convinced that this is synthetic biology is an entirely new way of thinking about and doing biology. And what distinguishes it is that it has an engineering orientation. We had genetic engineering in the past, but that was purely metaphorical. It's only now that we are in a position really to begin to think about engineering. And here you have a definition that's typical. It turns out there are scads of reports out there trying to get a handle on what synbio is and what its consequences are going to be, what its social outcomes are going to be. And they all have some attempt to offer some attempt to define the field, and here's one of them. Here's another very similar one. Here's one that's offered within the community itself on this website, syntheticbiology.org. The core idea here is to try to distinguish synthetic biology from analytic biology. Analytic biology was about understanding life. Synthetic biology is about building life and doing it in a rational way. Now, one problem with these definitions is that they're so broad that they're going to count a lot of things that maybe you don't really think of as synthetic biology. They don't really seem to count as synthetic biology. So if you go with a definition like this, perhaps the first instance of synthetic biology was the production of synthetic urea in a chemistry lab, which occurred in 1828. And people who offer these definitions that I've just thrown up are completely cool with that, and this is how they think about synthetic biology, and they see it as a thing that's been slowly developing starting in 1828. A narrower view would be, and this is basically sort of the working definition I'm going to go with here, that synthetic biology in its current form is about building microbes that will produce things we want or do things that we want. And typically, the building is going to mean modifying them genetically. But there's still no one stream of work going on in the field. Rather, there are a handful of different lines, different kinds of strategies. And there are various taxonomies that are offered in the literature. I'm going to offer you a version, a sort of a tweaked version of one developed by O'Malley and colleagues in 2007. Again, it was my colleague Eric Perrins that initially adopted this taxonomy and tweaked it, but as I've studied the field, I think this is quite right. So, first line. First, even if synthetic biology is different from genetic engineering of the last few decades, there's some of the work in the field that sure looks an awful lot like old-fashioned genetic engineering. The difference is just that before, scientists could switch out a gene or two. Now they can swap out entire suites of genes. They've got more information and they can much more powerful tools to do the work. The leading example of this work is stuff that Jay Kiesling, who's at UC Berkeley and now at DOE-funded enterprise, the Joint Bioenergy Institute out in California, undertook a few years ago. He figured out how to modify E. coli, which is what's pictured here, and then yeast, so that they would produce artemisinic acid. I think there's a cassette of about, cassette's not the word I want, but there's a suite of about 20 genes that he had to get to function within these cells. But once you've got the artemisinic acid extracted from these cells, then you can take it into the chemistry lab, and through three very simple sort of chemistry steps, you can turn it into artemisinin, which is a very effective, in fact, the last really effective line of defense against malaria. It's currently produced from wormwood, but the supply is limited. It's not adequate to meet the demand, and this offers a way of meeting what is, in fact, a huge global need. 500 million people a year get malaria. 1 million die each year from it. Now, I should say this is the part of the taxonomy I'm giving you which is the most controversial. Kiesling himself and O'Malley lump this work into this line. And this is the line that is really characterized by the engineering mindset. You could be doing advanced genetic engineering and think of biology as basically a semi-rational art, which is what a synthetic biologist I recently met said it is. He's one of the ones who's critical of this this stream. He sees it more as a kind of experimenting and playing. So here the idea is to design genetic sequences called in the field biotools or biobricks or bioparts that then you can assemble in different combinations to do different things you want. So you can actually, the idea is that they'll be like machines. They'll be interchangeable parts. They'll work together like the gears in this image, which is the logo of the BioBricks Foundation, or like Lego bricks. So you could have, for example, a sensor, little genetic sequence that codes for the capacity to detect a chemical in the environment, and then you could have an actuator that causes the cell to do something that you want, and you could have a transmitter that is tripped by the sensor and in turn gets the actuator going, and then you string them together and you've got a circuit, as they're called, and away you go. It often looks to me, following the conversations about this, that there's both a scientific and a social agenda at play here. The idea is you'll have radical simplification of genes. Each part will do a very, very particular thing and nothing else, and will always do exactly what you want. You'll have standardization of parts so that you can mix and match them in the way that I've described, and they'll be generally available to the public. So the BioBricks Foundation, which exists to promote this vision, is very active in advocating for an open access intellectual property framework. They want these things to be literally freely available to the public. And the idea eventually is that, laptop on the kitchen counter, as I was describing, you could go to a catalog and you could search for the parts you want to do thus and such a thing, and you could order them. This registry is an actually existing registry affiliated with MIT, and there are some thousands of parts that are now available. You can go and order. So the underlying idea here is that we're going to turn biology from science, dependent on very esoteric knowledge and a certain amount of ingenuity and good luck, into engineering. Something that'll be all very predictable and that you could actually do without really knowing all that much about the underlying science. So you could make new things out of microbes. You could create new microbes without actually knowing very much about the biological science. Part of this whole vision is the International Genetically Engineered Machines competition, annual competition that's been going on since 2004, in which undergraduate teams led by non-undergrads, people who know much more about what they're doing, try to design and build biobricks and then operate them in living cells. And then the biobricks that they create, one of the conditions for being part of this is that the biobrick will then be transferred over to the registry. You won't try to claim a patent on it. It'll be made publicly available. And again, there's sort of a social agenda here because not only are you actually advancing the work by trying to create biobricks, but you're sort of creating the incoming generation of people who are going to do this work and sort of winning people over to this whole way of thinking about biology. this image might look familiar if you were here for a talk that James King gave at a Garrison seminar, I think in spring of this year. The 2009 winner of iGEM was the Cambridge team. They produced bacterial biosensors that would respond to the presence of a chemical in the environment, and then produce various pigments. So all you had to do was see what color was produced in order to find out whether that chemical was in the environment, and perhaps what the concentration of the chemical was as well. And it's a kind of a cool thing. Tom Murray, president of the Hastings Center, who's not here tonight, by the way, because he's at the Presidential Commission for the Study of Bioethical Issues talking about synthetic biology down in Atlanta today. Tom Murray and I were at a workshop sponsored by the Sloan Foundation and DOE, and at one point Tom leaned over to me and suggested that we try to keep track of the number of times that synthetic biology was described as cool. And it would have been a very hard thing to do. But coolness notwithstanding, the success with the BioBricks model has been very limited to date, and my impression is that it's at best a very open question whether it's going to work out, and many in the field think it's not going to work out. And that's why in this taxonomy I've differentiated between advanced genetic engineering and this BioBricks line. It turns out that a lot of these bricks, even though they're available on the MIT registry or elsewhere, are not actually very well characterized, so you're not entirely sure what it is you're getting when you order it. And then when you assemble them in circuits, they work a little bit unpredictably depending on what parts you have them with, or maybe they turn out to be incompatible altogether with parts. You just can't mix and match them in the way you want. They're dependent upon the environment, which cell that you're putting them in, and so forth. Now, this is the line that's associated with the announcement that JCVI made in May, that they'd created a synthetic cell. Here the idea is to simplify not just parts, but cells themselves. JCVI has this project going which they call the Minimal Genome Project. They've started with another one of these mycoplasma varieties, Mycoplasma genitalium, which has one of the smallest known genomes of any microbe. It has about 500 genes. And then they've gone through and they've begun trying to strip out genes and figure out which ones turn out actually to be non-essential. So you can create a cell that has just the genes that you need to keep going. And then they've begun building around this work with a handful of other lines, a few of which I've got up on this slide. In 2007, they announced that they'd succeeded in taking a genome from a mycoides and sticking it into an empty capricolum and getting it to work. And then in 2008, they announced that they'd managed to chemically synthesize this very, very small variety of the mycoplasma, the genitalium. And then in 2010, they sort of put these two things together. They had synthesized a mycoides genome, and then they stuck it into this capricolum cell. So you can see this is sort of coming at cells from the other end, from the BioBricks. That's working on parts, this is working on cells, but the two lines are supposed to merge eventually. The idea is that this gives you sort of a platform or a chassis, a launching pad, that you can then stick BioBricks, bioparts into to make different kinds of machines. All right, so I said at the start that the JCVI announcement was billed as a huge technical accomplishment. So this is the point raise questions about that. My impression is that this work was nowhere near as path-breaking as what was often claimed. It's hard to see this really as a synthetic species, in spite of this small change that was made to the genome. It's hard to see it as a synthetic life form, or even really a synthetic cell. What this really is just a synthetic genome that's then been inserted into an existing cell species, Mycoplasma capricolum. So in the New York Times article about this work, when it came out, David Baltimore, Nobel laureate and biologist at Caltech, offered the thought that they hadn't really created life, synthesized life, so much as they had mimicked it. And maybe even better would be to say that they mimicked one part of it, the genome, and then they took advantage of some existing life in order to make the rest of it work. Now, the explanation from the Venter camp is that this is a synthetic cell because the genome was synthesized and the genome took over the cell. Once it was in the cell, it actually eventually redirected the development of the cell from the capricolum line into the mycoides line, and all of the parts of the cell were eventually replaced by these new mycoides parts. But this idea of a genome taking over a cell, this is a metaphor that is really relies on an assumption that cells have this sort of top-down organizational structure, that it's the genome that's really important about a cell, and everything else is just kind of following along. And this is a metaphor that is really widely accepted and very influential. Now, when the human genome was sequenced, we talked about finally having decoded the book of life. But there are a lot of biologists these days who question this metaphor. And if you reject that metaphor, then you might find yourself wondering, well, why say that it took over the cell? Why not say that the genome and the existing cell were joined and functioned? They turned out to function. Baltimore called this a technical tour de force. It's kind of a proof of principle that you can sequence a genome and you can get it into a cell, but it's not really breakthrough science. In a way, what was really surprising about this work was that it took them quite as long to get to this stage as it did, because they'd gotten the earlier steps in place many years earlier. This picture, by the way, is of these synthetic cells. And here's the great man himself, Craig Venter. If his name is familiar, it's because he was the one who was racing Francis Collins and the federal government to sequence the human genome. And the fourth line, this is the other way of going about creating minimal cells, and it's probably a better candidate for the label synthetic life. Here the idea is to try to abstract from all existing forms of life, identify the basic functions that are necessary to keep life going, and then to construct them maybe from scratch, maybe using entirely new materials. So in principle, you might end up with an entirely new biochemistry, one that's not carbon-based. You could have a non-organic biochemistry. This stream is often omitted in discussions of synthetic biology because many of those discussions just sort of assume that what's interesting about synthetic biology is the genetic stuff. If you're engaged in protocell creation, you might not be relying on genes as we understand them, and in fact, you might decide that replication turns out to be one of those parts of life that maybe is dispensable. Maybe what really counts about life is just that you have metabolism and control. this is a picture of some vesicles, artificial membranes, that were created by a company called Protolife that might be suitable for cells. These are not cells themselves. This is an installation at the Venice, I don't know how to pronounce this, Biennale, an architecture festival. It's supposed to be the leading architecture festival in the world. Canada's entry in this at the Canadian Pavilion was just written up recently in an essay in the science journal Nature. The author of the essay, who was involved in the design of Canada's entry, described this work that they displayed at the Biennale as a blending of artificial intelligence, synthetic biology, and interactive technology. It was called Hylozoic Ground. The synthetic biology angle here is that recycled carbon dioxide exhaled by visitors to the event is then transformed into solid carbonate using protocell technology. I checked this out with somebody I know who's working on protocell work, and he thought that this one should be chalked up mostly to hype. He said that these cells are not alive, and they're not even close to being the most lifelike protocellular achievements in the lab. But it is a beautiful image. So one of the recurring questions at SynBio conferences is what the killer apps are going to be. And just about everything under the sun has been envisioned at one point or another. There's a partial list here. Craig Venter has said that there is no field that he can envision will be untouched by synthetic biology. My colleague Jacob Moses was recently at a NASA conference on synthetic biology. They're trying to figure out how synthetic biology might be important in space travel. And one of the possibilities there was that we might engineer the human microbiome, the cells that are in the gut and living in your skin, so that humans could be made impervious to infection. And another possibility mentioned was that humans could be made so that they didn't have to eat on long space flight if we could only figure out how to make photosynthesizing human. Michael Spector, in that article in the New Yorker that I had a quote from earlier, quoted a few people in the field as proposing that we might finally get human reproduction under control. That instead of relying on chance combination of genes through sexual combination, that we could rationally design our children. It's also been proposed that we could make shelves out of wood wood cells that would just sort of grow into the shape that you want, or gourds that would grow into houses. All sorts of sort of bizarre architectural applications that have been envisioned. The article that mentioned Hylozoic Ground, that architectural exhibit from Canada, proposed that that same technology could be used to grow a limestone reef under Venice to keep it from sinking. Another possibility thrown out in that article is that we might grow pigskin cells to make a new kind of exterior cladding for buildings. And it was mentioned here that the biodegradable nature would avoid the need for later demolition. As a homeowner, I have a hard time actually picturing what the advantage is of avoiding the need for later demolition. And this actually can be done now, that growing pigskin cells to produce exterior cladding, but it is a little bit pricey just at the moment. It's about $1,000 for a 3 square centimeter patch, which puts it just out of my reach. Another application I thought I'd mention, it turns out it's possible to create cells that then you can seed into clouds where they'll take advantage of water vapor and ambient atmospheric chemicals and sunlight to produce foodstuffs. I don't want to overhype this. This is not something that's going to happen right away, but by and by. Some of the early attempts at this haven't panned out exactly as promised. These are my daughters with a blueberry pie that they made the old-fashioned way, grown organically in the field behind the porch there and picked by hand and baked in an oven. Hopefully we can get away from Luddite scenes like this in the future. But I don't want to I shouldn't leave you quite on this satirical note. There actually are some applications out there that are really well along. I mentioned the production of artemisinic acid. That is the real deal. That's there, as near as I can tell. There's also a tremendous amount of work that's going on different ways of producing biofuel, better ways of producing biofuel than we've got now. There are a handful of small startup companies out there relying on venture capital funding. Synthetic genomics, which is part of the Craig Venter empire, signed a many $100 million deal with ExxonMobil to investigate, see if they could figure out how to make algae that would produce fuel. And there's a major research initiative out in California, the Joint Bioenergy Institute. It's an interrelationship of three national laboratories and three major research universities. And they're all working on ways of producing biofuel essentially from wood. Is the idea. So it'd be much more effective than our current method, which is to produce it from sugar. This is an example that just happened to pop into the news in September. There was a story in the New York Times, I think it was in the business section. It's one of these little startup companies, Joule Unlimited, and the announcement in the story was that they'd won a patent on a blue-green algae, not technically an algae, a cyanobacterium, that supposedly uses photosynthesis to produce alkanes, hydrocarbon that's used in producing fuel. And they plan to begin construction next year on a commercial plant that would produce 15,000 gallons per year per acre. They claim that it would be quite a bit better than traditional biofuel. They claim that their method would be 50 times more efficient than the current ways of producing biofuel. And part of what's attractive about this, you get away from drilling for oil because you're producing it from scratch. You get away from having to transport it because you could produce it much closer to where it's being used. You wouldn't have to ship it overseas. And the method here is a photosynthetic method. You're not relying on a lot of land given over to producing feedstocks, like sugarcane or something, or wood. And the process, this photosynthetic process, relies on sunlight and carbon dioxide, so it's a carbon-fixing process. So in theory, you're also actually not only avoiding some of the environmental costs of producing oil, but you're offsetting some of the environmental costs of eventually burning it, because you're pulling carbon out of the atmosphere in order to produce the fuel. I ran this story by somebody I met recently who's out at JBay, the Joint Bioenergy Institute, and he said, well, it looks very capital intensive with all these little parts that they've got here. It looks like they haven't solved all of the problems, but they've made a lot of headway on some. One of the real issues in producing biofuel through synthetic biology means is you have to figure out some way of separating the fuel from the organism. But in this case, what they claim to be able to do is produce an organism that literally secretes the fuel as you go along. So that doesn't solve everything, but it's a huge step forward there. His take-home point, I think companies like Joule are going to be successful, but they won't be the entire solution, he went on to say. They'll be a part of the solution. And I should say, by way of full, giving the full story here, that this story went on and on with all these wonderful things, and then at the very, very end, it quoted an independent expert who said that they didn't yet have a proved concept. Now, so I'm not even entirely sure where that leaves them. They're planning to start, a production facility this year, but they haven't quite got what a venture capitalist would want to see. I don't know if this one really counts as synthetic biology, but certainly under some of the broader definitions that I was offering at the start, it would. As you may know, the production of hard cheese involves an enzyme called chymosin, which is found in rennet, which is produced from the stomachs of calves, unweaned calves. It's an enzyme that the calves use to process the mother's milk. It turns out that it's possible to take these cow genes and insert them into various other kinds of organisms, bacteria, fungi, yeast, and then produce the chymosin that way. Actually, this slide is inaccurate. It says modified yeast to rennet to chymosin. It would skip the rennet stage as well. It just produces the chymosin directly. And this was work that was done a decade or so ago, and it was approved by the FDA, and it's actually largely taken over the industry at this point. 80 to 90% of all the hard cheese that's produced in the United States relies on chymosin produced by genetically modified organisms. That's an interesting, I find this, frankly, a very challenging application. I found this particular image on a website that said, wine and cheese are said to be the purest and the most romantic link between humans and the earth. Well, so the question is, does this fact about how the cheeses are produced change your view of that? It's a very, very interesting question for me, and I'm not sure where I come down on it myself. So, when I was listing some of the possible applications, I said that synthetic biologists are always looking for the killer apps. Well, there's another way of thinking about that phrase, killer app. There are a few kinds of categories of risk. One large category has to do with ways that the technology could be deliberately misused. And this has already been proven possible. The 1918 Spanish influenza virus has been reproduced in the lab. Polio, which is an RNA virus, has been reproduced in the lab. Smallpox has not, but it will be. At some point it will be. And at some point it will probably be possible to go on and produce bacterial pathogens. And it might even be possible to produce new kinds of pathogens. There was some work done in Australia some years ago on mousepox, which is related, pretty closely related apparently to smallpox. And it was tweaked in a few ways that made it much more virulent, much more powerful killer than unaltered mousepox had been. And there'd be a variety of different kinds of targets for this. It wouldn't necessarily just be humans. Agricultural targets might be the most effective way of attacking a country. So this is clearly possible to do this kind of thing, but there's a little bit of debate about the likelihood of it. Some think, well, terrorists have better weapons at their disposal than this, you can't, these things you couldn't really control. They might end up hurting those who wielded them as much as the intended targets. On the other hand, one should probably never bank on the rationality of a terrorist. Another question about the likelihood of this actually being put into use is, it's not enough just to create the pathogen. You also have to have a way of weaponizing it. You have to have a way of distributing it. Well, I've heard synthetic biologists say, that's actually not that big a deal. All you need is 100 sort of versions of a suicide bomber, people who will infect themselves with something that is very, very contagious, and then go out into the city and go to movies and ride the subway, and you could have a real good epidemic going quite shortly. So that's one category of concern. Another is biosafety. If the first one was bioterrorism, then this one is bioerrorism, as it's been dubbed. Here the worry is about accidents, lab accidents, industrial accidents. Maybe the organisms will escape, they'll turn out in their new environment to have properties different from what we intended, different from what they had when they were in a contained environment. Maybe they'll mutate to acquire new properties, and then they could pose a threat, and again, the threat could be to public health, agriculture, environment. And the worry here is not just that the organisms will be dangerous, but that the genetic sequences within them could be dangerous. Supposing that they get out and don't succeed, they die, they spill their genetic contents. Those contents can easily be picked up by other microbes, and whatever properties they confer, and they could confer different properties in a new microbe, then might themselves be dangerous. But the debate mainly is about the microbes themselves somehow surviving in the wild. And of course, that accidents will occur is, again, this is proven, there's no debate about that. The debate, again, is about the severity of the risk. The synthetic biologists will tell you that the organisms that they're creating, especially if they're designed to be in a contained environment, like those panels that were featured in that slide by Joule Unlimited, those are not organisms that are designed to flourish in the wild. Those are organisms that are actually probably going to be too weak to survive in the wild. And they've been modified to do something that it doesn't actually benefit organisms to do in the wild. So they would die out really quite quickly. And two, it might be possible to design in fail-safe mechanisms to make sure that they die if they get out. And of course, fail-safe mechanisms always work. But in all fairness, this is exactly the kind of thing that synthetic biologists are trying to save us from. This is a catastrophe that we experienced in the midst of drilling for oil, and if we were able to effectively produce a lot of fuel by means of algae or blue-green algae, maybe this kind of thing could be made much less likely. Still, there are these concerns about very, very low probability, but potentially very high impact risks associated with synthetic biology, with bioterrorism and bioerrorism. A third category of concern, one I have a hard time myself really knowing quite what to make of. There are some who worry about very broad social and economic dislocation that might be caused by this new industrial revolution. If it's really a new industrial revolution, it's going to have major effects. Livelihoods are going to disappear. People are going to have to change. They're going to have to re-educate. And then what? The production of artemisinic acid. Well, one of the groups that's most ardently opposed to synthetic biology has said, right now, artemisinin is produced, it's extracted from wormwood, an herb pictured here, which is grown by some thousands of farmers, not a huge number, but farmers in developing countries. If you can successfully produce artemisinin using synthetic biology, then their livelihood completely disappears. And what about that? And biofuel. If the photosynthetic ways of producing biofuel, the Joule Unlimited strategy doesn't really pan out, and we end up with something from, say, the Joint Bioenergy Institute using wood or sugarcane, then you could end up with a huge demand in developed countries for biofuel that's been created this way. And there'd be a great impetus in developing countries to convert arable land from food production over to the production of biofuel for wealthy people in other countries. So that could be very damaging. That appears to me to be, I'm not quite, I have a hard time giving very much weight to the concern about wormwood growers, given the need there. But the concern about biofuel, that strikes me as a real concern. So I said at the beginning that different views of biotechnology often trace back to different stances about what the human relationship to nature should be. And actually, our primary charge in this Sloan-funded project, topic of some stuff I've written, and one of the things that I talked about when I was called to testify to the House Energy and Commerce Committee, this is a picture of that, and then again at the first meeting of the Presidential Commission for the Study of Bioethical Issues, was to think about this issue. There are issues that are basically consequentialist. They have to do with the outcomes, the risks, and the potential benefits. But you might also be concerned about synthetic biology on these intrinsic grounds, if you will. You might just see it as something that is in and of itself morally troubling. And so the question is, what to make of this kind of concern? Is that a legitimate kind of concern to have about synthetic biology, and could it lead to even to public policy restraining synthetic biology in some way? And in a nutshell, my conclusion that what I told the House was that yes, this is a legitimate moral point, but it's not going to turn out to be something that should generate restrictive policy. And this I'm offering is somebody who's attracted toward not attracted to the engineering kind of mindset. Well, someone who's attracted to a preservationist mindset. It seems to me that the challenge with this concern is to figure out how to articulate it. One way of developing it will be as a kind of religious, metaphysical, grounded concern. That we're doing something that is prohibited by God as we understand God's commands. And I think that's. I'm not going to argue with that, but I think that those kinds of concerns are not very easily incorporated into public policy. There's a gap there between concerns that are explicitly grounded on religious positions and public policy. And it actually turns out that whether people who come to synthetic biology from a religious background are opposed to it. The Catholic Church has officially responded to the JCVI announcement that a synthetic cell had been created by saying, wow, this looks like an interesting line of work. They did not come out against it. Now, there's some work done by the Woodrow Wilson Institute, which suggests that evangelical Christians are likelier to be just innately opposed to this line of work. If you develop this kind of concern not as grounded in religious positions, but just as a kind of moral point, and most of our moral arguments don't trace all the way back to metaphysical concerns. They're just objections that we try and get other people to see them the way we do. And many of us do have this kind of feeling about the natural world. We feel that species are to be protected and preserved, wildernesses are to be protected and preserved, and so forth. But here, I think that this isn't going to lead really to restrictive policy either, because the problems that you might envision if you have a preservationist mindset are going to be covered by the consequentialist concerns. If synthetic biology leads to environmental destruction, then that would be reason to have this intrinsic moral objection to it. But that kind of worry is also going to be covered by sort of straightforward concerns about public health. I wanted to say something about this picture. I gave testimony at the House, and then afterwards we were standing around. I had my bag on my shoulder and was about to leave, and this guy suddenly pointed at my hair and said, "I think you have a leaf in your hair." And I thought, oh, how perfect is that? It was my few minutes of fame, and I haven't even managed to comb my hair appropriately. And the idea that I would have a leaf in my hair seemed to be sort of in keeping with my own views about the moral concerns at stake. I also said at the beginning that the JCVI announcement was billed as not just as a technical advance, but as a huge philosophical advance. And the philosophical advance was supposed to be that it showed once and for all that vitalism, the philosophical position known as vitalism, had been proven wrong. Vitalism is the belief that living things aren't just material entities. They have some sort of non-material or spiritual thing going on in them as well, a soul or a spiritual essence. And what we now knew, according to folks at JCVI and some prominent bioethicists, including some friends of mine, was that this is wrong because we just created a microbe in the lab. So now we knew, nope, nothing special about life. Life is just a puddle of chemicals interacting in an organized way. But just as a matter of logic, this doesn't actually follow. If there's a God who breathes some sort of microbial soul into bacteria when they replicate, that God is also perfectly capable of breathing that soul into a microbe when it's created in the lab. There was actually a very analogous question arose when people were debating some years ago what had been established by making it possible to clone human beings. The question was, if you cloned a human being, would the human being have a soul? And it was actually sort of seriously proposed by a few people in bioethics that actually, no, they wouldn't have souls. But I think that's an extremely hard position to hold on to. If you have a person, moral being, responding to the world as we are, very much his or her own person, why wouldn't they have souls? Similarly, microbes created in the lab would seem to have whatever special thing microbes that arise naturally would have. But this got me thinking a little bit about, what would be the reaction to synthetic biology if it hadn't been hyped quite the way it was? If JCVI hadn't said, we've created life, we've created a synthetic organism. What if they just said, we've created a genome and stuck it into a cell and it's survived. You know, maybe there wouldn't have been so much of an outcry about it. And the other thing that I told the House is that we should be very careful about evaluating the outcomes, about weighing the risks and the potential benefits. And these are the recommendations, these first four are the recommendations I gave the House. In a way, they're kind of obvious recommendations. Pay more attention to the risks. Make sure that they're not just being evaluated by synthetic biologists. Try and bring other disciplines into play. Make sure that you're looking for unexpected outcomes. Make sure that the regulatory framework is adequate. In a way, I think what the interesting issue for me is whether the processes that we now have of studying outcomes are the right ones. Right now in our country, we often rely on a system that's basically a technical enterprise. We see outcomes evaluation as something to be done by committees of experts, by scientists and economists. But there are huge value questions at play. What counts as a risk? How great a risk is it? How heavily should you weigh it? How much should you discount a risk that will only be harmful at some point in the future? How much should you discount a risk if it's extremely unlikely? These are all value questions. And I think maybe synthetic biology gives us an opportunity to rethink the whole process of thinking about risks, because you're dealing with such weird risks in the case of synthetic biology. These low probability but high impact risks, which are so difficult to get your head around in the first place. So it seems to me that we need to make sure we're answering these questions in the way we want and conducting the process in a way that we can make sure our values are really brought to bear upon it. Thank you.